In this presentation, I'm going to clarify a few points on the amino acids topic of A22, specifically peptides, polypeptides, and proteins. Peptides, polypeptides, and proteins are all amino acids linked together. So from a chemistry point of view, we have to be able to look at how we put those together. And they're an example of a condensation reaction. And in fact, a protein is a condensation polymer. It's a long chain of individual molecules all joined together. And in CA examinations, you may be asked to show possible structures for some of the dipeptides where we put two amino acids together. And amino acids can be sometimes thought of as being analogous a little bit like to the letters in the alphabet. If you arrange 26 letters, you can get an infinite variety of words from that. And in the same way, when you've got 20 amino acids and you put them in different conformations, you can get an infinite variety of proteins. Now, the dipeptides, there are two possible structures each time you join two amino acids together. In the same way that if you had two letters and you put them in different orders, they form different words. So IS obviously is, and if you reverse those, SI becomes C, a Spanish word rather than an English word. So the dipeptide is the same idea. If we want to form two dipeptides, then what we'd do is we'd take our two amino acids and put them together. So looking at the screen, glycines on the left, alanines on the right, they link together by a condensation reaction. So for these to condense, that OH has to be lost from the acid group, the hydrogen from the amine group, and then they form the dipeptide with this water being eliminated. This region is the peptide bond, and that's the key part to adding amino acids together to make proteins and so on. The groups then, you'll see because glycine had this H group here, and then the difference with alanine is it's got a CH3. If you look from left to right, H on the left hand side, CH3 on the right. Now, the other dipeptide is basically reversing them around. So again, if you're looking at the screen, alanine's on the left, glycine on the right, and we put them together in exactly the same way with the elimination of a water molecule. Now, look at those two groups that we highlighted up a moment ago. CH3 this time is on the left, H is on the right. And what that means is if you're in an exam situation and you're asked to draw the two possible dipeptides for two amino acids being joined together, then you can do that simply by taking your time and drawing one structure, and then in the other structure just draw exactly the same but reverse those two variable groups. Below is an example for you to try. So alanine to serine. So use the structures that you're given there. You're given the condensed structural formula there for both alanine and for serine, and see if you can draw the two possible structures, and pause the video and try that, and then I'll give you the solutions in a moment. So the two possibilities, serine added to alanine, again serine on the left, alanine on the right, they join together with the elimination of water, and again you'll see the two groups there that we showed before, so that's the two variable groups. The other dipeptide would be alanine on the left, serine on the right, form the dipeptide with elimination of water, and you'll notice these two groups reversed. So if you link anything up to about 50 amino acids, that's termed a peptide or a polypeptide, and anything above that is usually termed a protein. To reverse that reaction that we've just seen, we formed the dipeptide by eliminating water, condensation reaction. So the reverse of an elimination reaction would be hydrolysis, water, hydrolysis, splitting. So using water to split the bond. So the peptide bond is that one, the C double bond O NH, and water basically would go in and break that bond. And the CO bit would become an acid again, COOH, and the NH bit would become an amine, NH2. To do that, we use fairly concentrated hydrochloric acid, about 6 molar, and you usually boil it, so probably under conditions of reflux, for about 24 hours, and that'll hydrolyze the dipeptide and break those peptide bonds and put them back to their amino acids. The structure of proteins you'll be familiar with from your biology. So 3 levels of structure. Primary structure is basically the sequence of amino acids, and if you think of it, they're joined by the peptide links. The secondary structure is the twisting or coiling of the chain. to form beta pleated sheets and alpha helices, and they're held together by hydrogen bonding. And then the tertiary structure is the bending or folding of that secondary structure to give a precise 3D shape, held together by hydrogen bonds, disulfide bridges, ionic interactions, and van der Waals forces. So if we look at those three structures, primary, as we said, is the sequence of amino acids. So there's an example of a sequence. That's probably the beginning of one, so that's why the space is left here. These are all joined together by the peptide bonds, the C double bond O NH link, and therefore that's going to be quite a strong chain. And then because each of them have got that NH and C double bond O, it's possible for hydrogen bonds to form between, for example, one amino acid and then one further down. So, for example, this alanine, the peptide link between this and leucine, could possibly form a hydrogen bond with, say, the bond down here for valine and whatever is linked to it. And what that starts to do, because that happens on a regular basis, you get a regular folding. So examples of this are the secondary structure. And the secondary structure you'll see in the diagram here. There's that peptide link. There's somewhere further along the chain, there's another peptide link, and you'll see the formation of a hydrogen bond between those. And because that happens on a regular basis, you get a regular structure. So in this case, a helix. You can also get an even more regular amino acids sequences. So for example, collagen or something like that, you could get a structure that actually ends up looking a bit more like a sheet. And you can see there superimposed in the back of that something that's supposed to look a wee bit like folded paper to emphasize that this is then what's called the beta pleated sheet, and this is the structure of the primary structure being folded into a regular secondary structure. And again, if you look there, you'll see the blue bits representing hydrogen bonds holding that together. So the secondary structure is held by hydrogen bonds. Primary structure by the peptide bonds, the secondary structure by the hydrogen bonds. Tertiary structure, well, that's the folding. There's various things that'll fold it. So if you've got an amino acid on the chain, it could be just by van der Waals forces attracted to another one, so that would fold the chain over. Or we've got the ionic interaction, so an NH3 group could be attracted to a COH group further along the chain, that could hold together. Or in some of the amino acids, they have SH groups on that side chain. And two SH groups that interact can bridge together using two sulfur atoms, and you get an S to S linkage. And that's what's sometimes called a disulfide bridge. Now, it's quite a strong interaction, and what it does is it folds the protein fairly firmly into a three-dimensional shape. So this picture looks as if it's a bit of random drawing and so on, but it's supposed to represent the tertiary structure of a protein. The sort of string is supposed to represent the primary structures, that would be the sequence of amino acids. You'll see every now and again nice regular helices. So there's a regular helices, there's an area of alpha helices, there's another area of alpha helices. And then these have been folded around, so perhaps some interaction between here and somewhere further along the chain. And then that folds into a definite shape. If you look at this, this is supposed to represent a substrate of an enzyme, and that substrate is only going to bond on if the correct chemical is here and here. So these, for particular enzymes or particular proteins, are actually very definite shapes, and they're held together by a variety of interactions. So ionic interactions, the van der Waals forces, the disulfide bridges, and also some hydrogen bonds as well. So this makes enzymes, and enzymes are basically biological catalysts. So any study of enzymes is similar to the study of catalysts. The difference is an enzyme is a protein that is a biological catalyst, and because of that, we have to think of the shapes of proteins and what affects those, and that's going to affect its ability to act as a catalyst. The enzyme as a catalyst has an active site, so that's where the substrate or the reactant would fit onto. And when a reaction involving an enzyme occurs, the substrate is turned into products. So in biology, they talk about substrate. We normally talk in chemistry about reactants, and they're turned into products. So it's basically the same process. The substrate is a slightly different term because it's quite often to do with fitting on to things like a protein. So there's a need for that substrate to be chemically equivalent. to something on the active site and allow it to be put in place. So it's complementary to the substrate. So the active site is the surface of the enzyme onto which the substrate fits. It tends to be a very small part of an enzyme. So there, for example, is a schematic representation of something like catalase. And catalase, as you'll see from your notes there, breaks hydrogen peroxide down into water and oxygen. Hydrogen peroxide would oxidize and therefore has to be removed from biological systems quite quickly. And so catalase is a very fast-acting enzyme. So you'll see that a large part of this is the main protein, and that would fold it into a particular shape, and then this part would be the active site onto which the hydrogen peroxide would fit. There's another, again, schematic representation showing an active site, and you can see the way it's quite often shown there as a particular shape, and then if you look at the substrate, it's a complementary shape, so that fits neatly into that. So how enzymes work is very similar to catalysis. So this diagram you'll have seen with catalysts. So reactants into products, an exothermic reaction, so energy being released. So there's delta H, the enthalpy of reaction going from the reactants down to the products. The activation energy without an enzyme present follows that red reaction profile. So for the reactants turning into products, you need quite a high activation energy. So at room temperature, very few of the reactants would have enough energy to turn into products. But when you add an enzyme, and what an enzyme does is by putting those molecules onto that active site, it could put stress onto the bonds that would lower the energy need to break those, or holding molecules closer together, so improving the orientation of the reaction. And what that does is it lowers the activation energy. Less energy is needed for that reaction to take place. So a higher proportion of the substances in the presence of an enzyme will have sufficient energy for a successful reaction. And you can see that lowers the activation energy and therefore works in a very similar way to a catalyst. The theory for this is what's called the induced fit theory. And again, you're probably familiar with that from your biology. So it means that the shape of a molecule, so the place where all of the atoms and so on are sitting, is not exactly the same as the shape of the enzyme. But as the substrate molecule starts to collide with an enzyme and come close to it, the interactions mean that the enzyme changes the shape very, very slightly, and then the substrate then fits onto it. And then you get an enzyme substrate complex forming, and that allows the reaction to be catalyzed. So here's again a schematic representation of it. You'll see the substrate here is supposed to be representing, although it's just a sort of squiggle line, so it's supposed to be representing the position of atoms and so on within the substrate. You can see that before the substrate is connected on, the active site is similar but not exactly the same as the shape of the substrate, so it doesn't fit exactly. But as it approaches and starts to make interactions there, you can see the change in the enzyme substrate, the enzyme shape on the active site, and the substrate fits perfectly into it. So the definition of induced fit is that the substrate induces a change in the shape of the active site of the enzyme. It can fit on, and then the reaction can be catalyzed. Finally, two things that affect enzyme activity: pH, and you've again probably familiar with this curve to show that they have an optimum pH under which they act. So this, for example, could be the curve for example, an enzyme in saliva, which is slightly alkaline. So this has got a peak at about 7.5, but look at the curve there. And you can see a small change in the pH greatly affects the activity of this. Now, the reason for this is if we go back to our shape of an enzyme, it's obviously going to be affected by all those interactions that hold it into the three-dimensional shape. So pH and the amount of H plus could affect ionic interactions, could affect hydrogen bonds. And so if you change the pH, you probably change the environment in which the enzyme is sitting, and its shape totally changes, and therefore the active site becomes wrong shape to fit the substrate on, and activity decreases. So they've got quite often a very specific pH range under which they act. And the other thing that gives them a narrow range is temperature. And you can see again from the shape of the curve there that this enzyme is reacting at a particular temperature, so 40 degrees centigrade. That ties in quite closely with sort of warm-bodied creatures and the temperature of warm biological systems. And as you start to move outside of that, you see that the activity decreases. Now, what's going to be happening when you change temperature is you're going to be affecting some of the bonds, you're going to be affecting the hydrogen bonds, you're going to affect some of the ionic interactions, and therefore, again, the conformation and shape of the enzyme is going to change. Temperature on the low side tends to have less of a dramatic effect. So if you look at that graph down here, about 10 or 5 degrees centigrade, you can see that the enzyme is still having some activity. So that's why, for example, your fridge will slow down reactions, but not stop them. But if you go to the other side, if you go to high temperature, that high temperature tends to completely break some bonds, maybe something like those disulfide bridges, so that the enzyme activity can't return again. And you'll see that at high temperatures, so up about 60, 70 degrees centigrade, quite often the enzyme activity completely crashes. And that's why, for example, we can sterilize by boiling. You break down the protein structure of things like bacteria and so on, and activity falls down.